The Art of Using t-SNE for Single-Cell Transcriptomics, Dmitry Kobik and Philip Barron's Nature Communications Volume 10, Article Number 5416, 2019. Cite this article. Abstract. Single-cell transcriptomics yields ever-growing data sets containing RNA expression levels for thousands of genes from up to millions of cells. Common data analysis pipelines include a dimensionality reduction step for visualizing the data in 2 dimensions, most frequently performed using t-distributed stochastic neighbor embedding, t-SNE. It excels at revealing local structure in high-dimensional data, but naive applications often suffer from severe shortcomings. For example, the global structure of the data is not represented accurately. Here we describe how to circumvent such pitfalls and develop a protocol for creating more faithful t-SNE visualizations. It includes PCA initialisation, a high learning rate, and multi-scale similarity kernels. For very large datasets, we additionally use exaggeration and downsampling-based initialisation. We use published single-cell RNA-seq datasets to demonstrate that this protocol yields superior results compared to the naive application of t-SNE. Similar content being viewed by others. Tutorial. Guidelines for annotating single-cell transcriptomic maps using automated and manual methods. Article May 24, 2021. Cross-platform normalization enables machine learning model training on microarray and RNA-seq data simultaneously. Article Open Access. February 25, 2023. Towards a comprehensive evaluation of dimension reduction methods for transcriptomic data visualization. Article Open Access. July 19, 2022. Introduction. Recent years have seen a rapid growth of interest in single-cell RNA sequencing, RNA-seq, or single-cell transcriptomics 1,2. Through improved experimental techniques, it has become possible to obtain gene expression data from thousands or even millions of cells, 3, 4, 5, 6, 7, 8. Computational analysis of such datasets often entails unsupervised exploratory steps, including dimensionality reduction for visualization. To this end, many studies today are using t-distributed stochastic neighbor embedding, or t-SNE9. This technique maps a set of high-dimensional points to 2 dimensions, such that ideally, close neighbors remain close and distant points remain distant. Informally, the algorithm places all points on the 2D plane, initially at random positions, and lets them interact as if they were physical particles. The interaction is governed by 2 laws. First, all points are repelled from each other. Second, each point is attracted to its nearest neighbors. See methods for a mathematical description. The most important parameter of t-SNE, called perplexity, controls the width of the Gaussian kernel used to compute similarities between points and effectively governs how many of its nearest neighbors each point is attracted to. The default value of perplexity in existing implementations is 30 or 50, and the common wisdom is that the performance of t-SNE is fairly robust to changes in the perplexity. 9. When applied to high-dimensional but well-clustered data, t-SNE tends to produce a visualization with distinctly isolated clusters, which often are in good agreement with the clusters found by a dedicated clustering algorithm. This attractive property, as well as the lack of serious competitors until very recently 10,11, made t-SNE the de facto standard for visual exploration of RNA-seq data. At the same time, t-SNE has well-known but frequently overlooked weaknesses 12. Most importantly, it often fails to preserve the global geometry of the data. This means that the relative position of clusters on the t-SNE plot is almost arbitrary and depends on random initialization more than on anything else. While this may not be a problem in some situations, RNA-seq datasets often exhibit biologically meaningful hierarchical structure, for example, encompass several very different cell classes, each further divided into various types. Typical t-SNE plots do not capture such global structure, yielding a suboptimal and potentially misleading visualization. In our experience, the larger the dataset, the more severe this problem becomes. Other notable challenges include performing t-SNE visualizations for very large datasets, for example, 1 million of cells or more, or mapping cells collected in follow-up experiments onto an existing t-SNE visualization. Here we explain how to achieve improved t-SNE visualizations that preserve the global geometry of the data. Our method relies on providing PCA initialization, employing multi-scale similarities 13,14, increasing the learning rate 15, and for very large data sets, additionally using the so-called exaggeration and downsampling-based initialization. To demonstrate these techniques, we use several full-length and UMI-based data sets with up to 2 million cells, Table 1. We use fit-SNE16, a recently developed fast t-SNE implementation for all experiments. Table 1. Data sets used in this study, listed in the order of appearance in the text. In all cases, we relied on quality control and clustering performed in the original publications. For the 10x genomics dataset, we used cluster labels from Ref 23, full-size table. In many challenging cases, our t-SNE pipeline yields visualizations that are better than the state-of-the-art. We discuss its advantages and disadvantages compared to UMAP10, a recent dimensionality reduction method that is gaining popularity in the RNA-seq community. 11. We also describe how to position new cells on an existing t-SNE reference atlas, and how to visualize multiple related datasets side by side in a consistent fashion. We focus on single-cell transcriptomics, but our recommendations are more generally applicable to any dataset that has hierarchical organization, which is often the case, for example in single-cell flow or mass cytometry 17,18, whole genome sequencing 19,20, as well as outside of biology 21. Results: preserving global geometry with t-SNE. To illustrate that the default t-SNE tends to misrepresent the global geometry, we first consider a toy example, Figure 1. This synthetic dataset consists of points sampled from 1550 dimensional spherical Gaussian distributions, grouped into 3 distinct and non-overlapping classes. The data are generated such that the types within 2 classes, and per type respectively, do not overlap, and the types within the 3rd class, per type, are partially overlapping. As a result, this dataset exhibits hierarchical structure typical for RNA-seq data. Figure 1. Figure 1. Synthetic dataset. The points were sampled from a mixture of 1550 dimensional Gaussian distributions. Total sample size n equals 15,500. A multidimensional scaling of 15 class means. Point sizes are proportional to the number of points per class. B. The first 2 principal components of the data. Point colors denote class membership, K and N. Nearest neighbor preservation, K and C. Nearest classes preservation, CPD. Spearman correlation between pairwise distances. C. Default t-SNE with perplexity 30, random initialization, and learning rate 200. D. t-SNE with perplexity. E. t-SNE with PCA initialization. F. t-SNE with multi-scale similarities. Perplexity combination of 30 and PCA initialization and learning rate. Full-size image. 2 classical methods to visualize high-dimensional data are multidimensional scaling, MDS, and principal component analysis, PCA. MDS is difficult to compute with a large number of points, here n equals 15,500, but can be easily applied to class means, clearly showing the 3 distinct classes, Figure 1a. PCA can be applied to the whole data set and demonstrates the same large-scale structure of the data, Figure 1b, but no within-class structure can be seen in the first 2 PCs. In contrast, t-SNE clearly shows all 15 types, correctly displaying 10 of them as fully isolated and 5 as partially overlapping, Figure 1c. However, the isolated types end up arbitrarily placed, with their positions mostly depending on the random seed used for initialization. In order to quantify numerically the quality or faithfulness of a given embedding, we used 3 different metrics, K and N. The fraction of nearest neighbors in the original high-dimensional data that are preserved at nearest neighbors in the embedding 22. We used and computed the average across all points KNN quantifies preservation of the local or microscopic structure, KNC, the fraction of nearest class means in the original data that are preserved as nearest class means in the embedding. This is computed for class means only and averaged across all classes. For the synthetic dataset we used, and for the real datasets analyzed below we used, the KNC quantifies preservation of the mesoscopic structure, CPD, Spearman correlation between pairwise distances in the high-dimensional space and in the embedding 11. Computed across all 499,500 pairs among 1,000 randomly chosen points, CPD quantifies preservation of the global or macroscopic structure. Applying these metrics to the PCA and t-SNE embeddings, Figure 1bc, shows that t-SNE is much better than PCA in preserving the local structure, KNN 0.13 versus 0.00, but much worse in preserving the global structure, KNC 0.23 versus 1.00, and CPD 0.51 versus 0.85. Our recipe for a more faithful t-SNE visualization is based on three ideas that have been previously suggested in various contexts: multi-scale similarities 13,14, PCA initialization, and increased learning rate 15. Figure 1c used perplexity 30, which is the default value in most t-SNE implementations. Much larger values can yield qualitatively different outcomes. As large perplexity yields longer-ranging attractive forces during t-SNE optimization, the visualization loses some fine detail but pulls larger structures together. As a simple rule of thumb, we take 1% of the sample size as a large perplexity for any given dataset. This corresponds to perplexity 155 for our simulated data and results in 5 small clusters belonging to the same class attracted together. Figure 1d. Our metrics confirmed that, compared to the standard perplexity value, the local structure, KNN, deteriorates, but the global structure, KNC and CPD, improves. A multi-scale approach, using multiple perplexity values at the same time, has been previously suggested to preserve both local and global structure, 13,14. We adopt this approach in our final pipeline and, whenever, combine perplexity 30 with the large perplexity. See below. Separate evaluation not shown here. Another approach to preserve global structure is to use an informative initialization, for example the first 2 PCs, after appropriate scaling, see methods. This injects the global structure into the t-SNE embedding, which is then preserved during the course of t-SNE optimization while the algorithm optimizes the fine structure, Figure 1e. Indeed, KNN did not depend on initialization, but both KNC and CPD markedly improved when using PCA initialization. PCA initialization is also convenient because it makes the t-SNE outcome reproducible and not dependent on a random seed. The third ingredient in our t-SNE protocol is to increase the learning rate. The default learning rate in most t-SNE implementations is, which is not enough for large datasets and can lead to poor convergence and/or convergence to a suboptimal local minimum 15. A recent Python library for RNA-seq analysis, Scampi, increased the default learning rate to 100,023, whereas Ref 15 suggested to use. We adopt the latter suggestion in use whenever it is above 200. This does not have a major influence on our synthetic dataset because its sample size is not large enough for this to matter, but will be important later on. Putting all three modifications together, we obtain the visualization shown in Figure 1f. The quantitative evaluation confirmed that in terms of the mesoscopic/macroscopic structure, our suggested pipeline strongly outperformed the default t-SNE and was better than large perplexity or PCA initialization on their own. At the same time, in terms of the microscopic structure, it achieved a compromise between the small and the large perplexities. Faithful t-SNE of transcriptomic datasets. To demonstrate these ideas on a real-life dataset, we chose to focus on the dataset from Tosich et al. 3. It encompasses 23,822 cells from adult mouse cortex, split by the authors into 133 clusters with strong hierarchical organization. Here and below, we used a standard pre-processing pipeline consisting of sequencing depth normalization, feature selection, log transformation, and reducing the dimensionality to 50 PCs. See methods. In the t-SNE et al. Data, 3 well-separated groups of clusters are apparent in the MDS, Figure 2a, and PCA, Figure 2b, plots, corresponding to excitatory neurons, cold colors, inhibitory neurons, warm colors, and non-neural cells such as astrocytes or microglia, gray/brown colors. Performing PCA on these 3 data subsets separately, supplementary Figure 1, reveals further structure inside each of them. For example, inhibitory neurons are well separated into 2 groups, PVOB/SST expressing red/yellow, and VIP/LAMP5 expressing purple/salmon, as can also be seen in Figure 2a. This demonstrates the hierarchical organization of the data. Figure 2. Figure 2. t-SNE et al. Dataset. Sample size. The cluster assignments and cluster colors are taken from the original publication 3. Warm colors correspond to inhibitory neurons, cold colors correspond to excitatory neurons, brown/gray colors correspond to non-neural cells. MDS on class means. Point sizes are proportional to the number of points per class. B. The first 2 principal components of the data. K and N. 10 nearest neighbor preservation, KNC. 10 nearest classes preservation, CPD. Spearman correlation between pairwise distances. C. Default t-SNE with perplexity 30, random initialization, and learning rate 200. D. t-SNE with perplexity. The labels denote large groups of clusters. E. t-SNE with PCA initialization. F. t-SNE with multi-scale similarities. Perplexity combination of 30 and PCA initialization and learning rate. Full-size image. This global structure is missing from a standard t-SNE visualization, Figure 2c. Excitatory neurons, inhibitory neurons, and non-neural cells are all split into multiple islands that are shuffled among each other. For example, the group of purple clusters, VIP interneurons, is separated from a group of salmon clusters, a closely related group of LAMP5 interneurons, by some excitatory clusters, misrepresenting the hierarchy of cell types. This outcome is not a straw man. The original paper 3 features a t-SNE figure, qualitatively very similar to our visualization. Perplexity values in the common range, for example 20, 50, 80, yield similar results, confirming that t-SNE is not very sensitive to the exact value of perplexity. In contrast, setting perplexity to 1% of the sample size, in this case to 238, pulls together large groups of related types, improving the global structure. KNC and CPD increase, at the expense of losing some of the fine structure. KNN decreases, Figure 2d. PCA initialization with default perplexity also improves the global structure. KNC and CPD increase, compared to the default t-SNE, Figure 2e. Finally, our suggested pipeline with multi-scale similarities, perplexity combination of 30 and PCA initialization, and learning rate. Yields an embedding with high values of all 3 metrics, Figure 2f. Compared to the default parameters, these settings slowed down fit-SNE from 30s to 2 meters, which we still find to be an acceptable runtime. It is instructive to study systematically how the choice of parameters influences the embedding quality, Figure 3. We found that the learning rate only influences KNN. The higher the learning rate, the better preserved is the local structure until it saturates at around. Figure 3a. In agreement with the results of Ref 15. The other 2 metrics, KNC and CPD, are not affected by the learning rate, Figure 3ce. The perplexity controls the trade-off between KNN and KNC. The higher the perplexity combined with 30, the worse is the microscopic structure, Figure 3b, but the better is the mesoscopic structure, Figure 3d. Our choice of provides a reasonable compromise. Finally, The PCA initialization strongly improves the macroscopic structure as measured by the CPD, Figure 3EF, while the other two parameters have little influence on it. Figure 3. Figure 3. The influence of parameter values on embedding quality. All panels show quality assessments of various t-SNE embeddings of the Tosich et al. Dataset. Attend nearest neighbors preservation, KNN, as a function of learning rate. Black line shows PCA initialization. Gray lines show random initializations with 3 different random seeds. Large black dot denotes our preferred parameter values. Perplexity combination of 30 and .B10 nearest neighbors preservation as a function of perplexity used in combination with perplexity 30. Learning rate. .CD. The same for 10 nearest classes preservation, KNC. The same for Spearman correlation between pairwise distances, CPD. Full-size image. To demonstrate that our approach is equally well applicable to UMI-based transcriptomic data, we considered 3 further datasets. First, we analyzed the N equals 44,808 mouse retina data set from Ref 24. Our t-SNE result preserved much of the global geometry, Figure 4a. For example, multiple amacrin cell clusters, green, bipolar cell clusters, blue, and non-neural clusters, magenta, were placed close together. The t-SNE analysis performed by the authors in the original publication relied on downsampling and had a worse representation of the cluster hierarchy. Figure 4. Figure 4. UMI-based datasets. Cluster assignments and cluster colors are taken from the original publications. Macosco et al. 24. N equals 44,808 cells from the mouse retina. Bipolar cells comprise 8 clusters. Amacrin cells comprise 21 clusters. Non-neural clusters are abbreviated Mg Muller glia, A astrocytes, F fibroblasts, P parasites, E endothelium, M microglia. B Shakar et al. 25. N equals 27,499 cells from the mouse retina, mostly bipolar cells. BC bipolar cell, RBC rod bipolar cell. Putative doublets contaminants shown in gray, yellow, rod and cone photoreceptors, cyan, amacrin cells. Some clusters appear to consist of 2 parts. This is due to an experimental batch effect that we did not remove. C. Harris et al. 26. N equals 3,663 hippocampal interneurons. Circles denote cluster centroids. The centroid of one cluster, SST Cryab, is not shown because its cells were scattered all over the embedding, as they did in the original publication as well. Cluster labels not shown for visual clarity. Full-size image. Second, we analyzed the N equals 27,499 dataset from Ref 25 that sequenced cells from mouse retina targeting bipolar neurons. Here again, our t-SNE result, Figure 4b, is consistent with the global structure of the data. For example, off bipolar cells, types 1 to 4, warm colors, and on bipolar cells, types 5 to 9, cold colors, are located close to each other, and 4 subtypes of type 5 are also close together. This was not true for the t-SNE shown in the original publication. This dataset shows one limitation of our method. The data contains several very distinct but very rare clusters, and those appear in the middle of the embedding instead of being placed far out on the periphery. See discussion. Finally, we analyzed a dataset of hippocampal interneurons from Ref 26. The original publication introduced a novel clustering and feature selection method based on the negative binomial distribution and used a modified negative binomial t-SNE procedure. Our t-SNE visualization, Figure 4c, did not use any of that but nevertheless led to an embedding very similar to the one shown in the original paper. Note that for data sets of this size, our method uses perplexity and learning rate that are close to the default ones. Positioning new points on an existing t-SNE atlas. A common task in single-cell transcriptomics is to match a given cell to an existing reference dataset, for example, introducing a protocol called patch-seq. Ref 27 performed patch clamp electrophysiological recordings, followed by RNA sequencing of inhibitory cells in layer 1 of mouse visual cortex. Given the existence of the much larger t-SNE et al. data set described above, it is natural to ask where on the figure 2F, taken as a reference atlas, these patch-seq cells should be positioned. It is often claimed that t-SNE does not allow out-of-sample mapping, i.e., no new points can be put on a t-SNE atlas after it is constructed. What this means is that t-SNE is a non-parametric method that does not construct any mapping. From a high-dimensional to the low-dimensional space, parametric t-SNE is possible but is out of scope of this paper, see discussion. Nevertheless, there is a straightforward way to position a new, on an existing t-SNE atlas. For each Cadwell et al. Cell, we found its nearest neighbors among the t-SNE et al. reference cells using Pearson correlation across the log transform counts of the most variable t-SNE et al. Genes as distance 28. Then we positioned the cell at the median t-SNE location of these reference cells, figure 5a. The result agreed very well with the assignment of Cadwell et al. Cells to the t-SNE et al. Clusters performed in Ref 3. Figure 5. Figure 5. Out-of-sample mapping. Interneurons from Ref 27 positioned on the reference t-SNE Atlas 3 from Figure 2F. Only VIP/LAMP5 continent from Figure 2F is shown here, as no cells mapped elsewhere. Cluster labels are given only for clusters where at least one cell mapped to NGC neurogliaform cells, SBC single bouquet cells. 2 cells out of 46 are not shown because they had an ambiguous class assignment. A cross-validation approach, 100 random VIP/LAMP5 cells from the t-SNE et al. data set were removed from the t-SNE embedding and then positioned back on it using our method. Black dots show new positions, black lines connect them to the original t-SNE locations of the same cells. Positioning uncertainty for several exemplary cells from Panel A. Polygons are convex holes covering 95% of bootstrap repetitions. Full-size image. An important caveat is that this method assumes that for each new cell, there are cells of the same type in the reference dataset. Cells that do not have a good match in the reference data can end up positioned in a misleading way. However, this assumption is justified whenever cells are mapped to a comprehensive reference atlas covering the same tissue, as in the example case shown here. In a more sophisticated approach, 24, 29, 30, each new cell is initially positioned as outlined above, but then its position is optimized using the t-SNE loss function. The cell is attracted to its nearest neighbors in the reference set, with the effective number of nearest neighbors determined by the perplexity. We found that the simpler procedure without this additional optimization step worked well for our data. The additional optimization usually has only a minor effect, 30. We can demonstrate the consistency of our method by a procedure similar to a leave one out cross-validation. We repeatedly removed one random t-SNE et al. Cell from the VIP/LAMP5 clusters and positioned it back on the same reference t-SNE atlas, excluding the same cell from the nearest neighbors. Across 100 repetitions, the average distance between the original cell location and the test positioning will mean SD, see Figure 5b for a scale bar, and most test cells stayed within their clusters. Positioning uncertainty can be estimated using bootstrapping across genes, inspired by Ref 3. For each of the patch-seq cells, we repeatedly selected a bootstrapped sample from the set of highly variable genes and repeated the positioning procedure 100 times. This yielded a set of bootstrapped mapping locations. The larger the variability in this set, the larger the uncertainty. To visualize the uncertainty, we show a convex hole covering 95% of the bootstrap repetitions, Figure 5c, which can be interpreted as a 2D confidence interval. A large polygon means high uncertainty. A small polygon means high precision. For some cells, the polygons are so small that they are barely visible in Figure 5c. For some other cells, the polygons are larger and sometimes spread across the border of 2 adjacent clusters. This suggests that the cluster assignments for these cells are not certain. Aligning 2 t-SNE visualizations. Tausig et al. 3 is a follow-up to Tausig et al. 31 where cells from mouse visual cortex were sequenced with an earlier sequencing protocol. If one excludes from the new data set all clusters that have cells mostly from outside of the visual cortex, then the remaining data set has cells. How similar is the cluster structure of this newer and larger data set compared to the older and smaller one? One way to approach this question is through aligned t-SNE visualizations. To obtain aligned t-SNE visualizations, we first perform t-SNE of the older dataset 31 using PCA initialisation and perplexity 50, figure 6a. We then position cells of the newer dataset 3 on this reference using the procedure described above and use the resulting layout as initialization for t-SNE with learning rate and perplexity, combination of 30 and, as elsewhere, the resulting t-SNE embedding is aligned to the previous one, figure 6b. Figure 6. Figure 6. Aligned embeddings. A t-SNE visualization of the data set from Ref 31. Cluster assignments and cluster colors are taken from the original publication. Circles show cluster centroids. Numbers highlight some noteworthy cases, see text. B t-SNE visualization of the data set from Ref 3, after excluding all clusters that mostly consisted of cells from anterior lateral motor cortex, 23 clusters that had ALM in the cluster name. This t-SNE analysis was initialized by positioning all cells on the reference atlas from panel A, ensuring that the two panels are aligned with each other. Full-size image. Several observations are highlighted in figure 6. 1 and 2 are examples of well-isolated clusters in the 2016 data that remained well-isolated in the 2018 data. SS de Chodl and Pvalb Viper 2. Here and below we use the 2018 nomenclature. It is an example of a small group of cells that was not assigned to a separate cluster back in 2016, became separate on the basis of the 2018 data, but in retrospect appears well-isolated already in the 2016 t-SNE plot. 2L5 LP VISP clusters. Finally, 4 shows an example of several clusters in the 2016 data merging together into one cluster based on the 2018 data, L4 IT VISP 1. These observations are in good correspondence with the conclusions of Ref 3, but we find that t-SNE adds a valuable perspective and allows for an intuitive comparison. Performing t-SNE on large datasets. Large datasets with present several additional challenges to those already discussed above. First, vanilla t-SNE 9 is slow for and computationally unfeasible for. See methods. A widely used approximation called Barnes-Hutt t-SNE 32 in turn becomes very slow for. For larger datasets, a faster approximation scheme is needed. This challenge was effectively solved by Ref 16, who developed a novel t-SNE approximation called fit-SNE, based on an interpolation scheme accelerated by the fast Fourier transform. Using fit-SNE, we were able to process a dataset with 1 million points and 50 dimensions, perplexity 30, in 29 min on a computer with 43.4 GHz double-threaded cores, and in 11 min on a server with 22.2 GHz double-threaded cores. Second, 4. t-SNE with default optimization parameters tends to produce poorly converged solutions and embeddings with continuous clusters fragmented into several parts. Various groups 16,23 have noticed that these problems can be alleviated by increasing the number of iterations, the length or strength of the early exaggeration, see methods, or the learning rate. Ref 15 demonstrated in a thorough investigation that dramatically increasing the learning rate from the default value, 2, where 12 is the early exaggeration coefficient 33, prevents cluster fragmentation during the early exaggeration phase and yields a well-converged solution within the default 1000 iterations. Third, 4. t-SNE embeddings tend to become very crowded, with little white space even between well-separated clusters, 18. The exact mathematical reason for this is not fully understood, but the intuition is that the default perplexity becomes too small compared to the sample size, repulsive forces begin to dominate, and clusters blow up and coalesce like adjacent soap bubbles. While so far there is no principled solution for this in the t-SNE framework, a very practical trick suggested by Ref. 34 is to increase the strength of all attractive forces by a small constant exaggeration factor between 1 and 10. See methods. This counteracts the expansion of the clusters. Fourth, our approach to preserve global geometry relies on using large perplexity and becomes computationally unfeasible because fit-SNE runtime grows linearly with perplexity. For such sample sizes, the only practical possibility is to use perplexity values in the standard range 10 to 100. To address this challenge, we make an assumption that global geometry should be detectable even after strong downsampling of the dataset. This suggests the following pipeline. I. Downsample a large dataset to some manageable size. 2. Run t-SNE on the subsample using our approach to preserve global geometry. 3. Position all the remaining points on the resulting t-SNE plot using nearest neighbors. 4. Use the result as initialization to run t-SNE on the whole dataset. We demonstrate these ideas using two currently largest RNA-seq datasets. The first one is a 10x genomics dataset with cells from mouse embryonic brain. We first created a t-SNE embedding of a randomly selected subset of cells, figure 7a. As above, we used PCA initialization, perplexity combination of 30 and learning rate. We then positioned all the remaining cells on this t-SNE embedding using their nearest neighbors. Here we used Euclidean distance in the PCA space, and, as above, this took 10 min. Finally, we used the result as initialization to run t-SNE on all points using perplexity 30, exaggeration coefficient 4, and learning rate. Figure 7b. Figure 7. Figure 7. 10x genomics dataset. Sample size. Cluster assignments and cluster colors are taken from Ref 23, a t-SNE of a random subsample of cells, PCA initialization, perplexity combination of 30 and 250, learning rate. The cluster labels for several small clusters, 30, 35, 36, and 38, are not shown here and in B because these clusters were very dispersed in the embeddings. t-SNE of the full dataset. All cells were positioned on the embedding in panel A, and this was used as initialization. Perplexity 30, exaggeration 4, learning rate. C. The same as in B, but without exaggeration. D. The same as in B, but with PCA initialization, i.e., without using the downsampling step. E. Default t-SNE with learning rate set to random initialization, no exaggeration. Full-size image. To validate this procedure, we identified meaningful biological structures in the embedding using developmental marker genes 35, 36, 37. The left part of the main continent is composed of radioglial cells expressing ALDOC and SLC1A3, figure 8a. The neighboring areas consist of neural progenitors, neuroblasts, expressing ohms, previously known as TBR2, figure 8b. The right part of the main continent consists of mature excitatory neurons expressing pan-neuronal markers such as SDMN2 and TUB3, figure 8c, but not expressing inhibitory neuron markers GAD1 or GAD2, figure 8d, whereas the upper part of the embedding is occupied by several inhibitory neuron clusters, figure 8d. This confirms that our t-SNE embedding shows meaningful topology and is able to capture the developmental trajectories from radioglia to excitatory/inhibitory neural progenitors to excitatory/inhibitory mature neurons. Figure 8. Figure 8. Developmental marker genes. Overlay over t-SNE embeddings from Figure 7. Expression of ALDOC gene, marker of radioglia, on the t-SNE embedding from Figure 7b. Any cell with ALDOC detected, UMI count above 0, was colored in red. Another radioglia marker, SLC1A3, had similar but a bit broader expression. B. Expression of ohms, marker of neural progenitors, neuroblasts. C. Expression of SDMN2, marker of mature neurons. A pan-neuronal marker TUB3 had similar but a bit broader expression. D. Expression of GAD1 and GAD2, either of them, markers of inhibitory neurons. EH. The same genes overlaid over the default t-SNE embedding from Figure 7e. Full-size image. We illustrate the importance of the components of our pipeline by a series of control experiments. Omitting exaggeration yielded overexpanded clusters and less discernible global structure, Figure 7c. Without downsampling, the global geometry was preserved worse, Figure 7d. For example, most of the interneuron clusters are in the lower part of the figure, but clusters 17 and 19, developing interneurons, are located in the upper part. Finally, the default t-SNE with random initialisation and no exaggeration, but learning rate set to, yielded a poor embedding that fragmented some of the clusters and misrepresented global geometry, Figure 7e. Indeed, overlaying the same marker genes showed that developmental trajectories were not preserved, and related groups of cells, for example interneurons, were dispersed across the embedding, Figure 8EH. Again, this is not a straw man. This embedding is qualitatively similar to the one shown in the literature, 23,38. In addition, we analyzed a dataset encompassing cells from mouse embryo at several developmental stages. 8. The original publication showed a t-SNE embedding that we reproduced in Figure 9a. Whereas it showed a lot of structure, it visibly suffered from all the problems mentioned above. Some clusters were fragmented into parts. For example, clusters 13 and 15, there was little separation between distinct cell types, and global structure was grossly misrepresented. The authors annotated all clusters and split them into 10 biologically meaningful developmental trajectories. These trajectories were intermingled in their embedding. In contrast, our t-SNE embedding, Figure 9b, neatly separated all 10 developmental trajectories and arranged clusters within major trajectories in a meaningful developmental order. For example, there was a continuous progression from radial glia, cluster 7, to neural progenitors, 9, to postmitotic premature neurons, 10, to mature excitatory and inhibitory 15 neurons. Figure 9. Figure 9. Cao et al. Dataset. Sample size. Cluster assignments and cluster colors are taken from the original publication. 8. A t-SNE embedding from the original publication. The authors ran t-SNE in Scampi with default settings, i.e. with random initialization, perplexity 30, and learning rate 1000. For cluster annotations, see original publication. B. t-SNE embedding produced with our pipeline for large datasets, a random sample of cells was embedded using PCA initialization, learning rate, and perplexity combination of 30 and 250. All other cells were positioned on resulting embedding, and this was used to initialize t-SNE with learning rate, perplexity 30, and exaggeration 4. Labels correspond to the 10 developmental trajectories identified in the original publication. Labels in capital letters denote trajectories consisting of multiple clusters. 32,011 putative doublet cells are not shown in either panel. Full-size image. Comparison with UMAP. A promising dimensionality reduction method called UMAP-10 has recently attracted considerable attention in the transcriptomics community. 11. Technically, UMAP is very similar to an earlier method called LargeVis39, but Ref10 provided a mathematical foundation and a convenient Python implementation. LargeVis and UMAP use the same attractive forces as t-SNE does, but change the nature of the repulsive forces and use a different sampling-based approach to optimization. UMAP has been claimed to be faster than t-SNE and to outperform it in terms of preserving the global structure of the data. 10,11. While UMAP is indeed much faster than Barnes-Hut t-SNE, FIT-SNE 16 is at least as fast as UMAP. We found FIT-SNE 1.1 with default settings to be 4 times faster than UMAP 0.3 with default settings when analyzing the 10x genomics, 14 meters versus 56 meters, and the Cao et al. 31M versus 126M datasets on a server with 22.2 GHz double-threaded cores. For this experiment, the input dimensionality was 50 and the output dimensionality was 2. UMAP may be more competitive in other settings. That said, the exact runtime will depend on the details of implementation, and both methods may be further accelerated in future releases or by using GPU parallelization 40. To compare UMAP with our t-SNE approach in terms of preservation of global structure, we first ran UMAP on the synthetic and on the t-SNE et al. 3 datasets, supplementary figure 2. We used the default UMAP parameters and also modified the two key parameters, number of neighbors and tightness of the embedding, to produce a more t-SNE-like embedding. In both cases and for both datasets, all three metrics, KNN, KNC, and CPD, were considerably lower than with our t-SNE approach. Notably, we observed that in some cases, the global structure of UMAP embeddings strongly depended on the random seed. Next, we applied UMAP with default parameters to the 10x genomics and the Cao et al. Datasets. Here, UMAP embeddings were qualitatively similar to our t-SNE embeddings, but arguably misrepresented some aspects of the global topology. Supplementary figure 3. An in-depth comparison of t-SNE and UMAP is beyond the scope of our paper, but this analysis suggests that previous claims that UMAP vastly outperforms t-SNE 11 might have been partially due to t-SNE being applied in a suboptimal way. Our analysis also indicates that UMAP does not necessarily solve t-SNE's problems out of the box and might require as many careful parameter and/or initialization choices as t-SNE does. Many recommendations for running t-SNE that we made in this manuscript can likely be adapted for UMAP. Discussion. The fact that t-SNE does not always preserve global structure is one of its well-known limitations. 12. Indeed, the algorithm, by construction, only cares about preserving local neighborhoods. We showed that using informative initialization, such as PCA initialization or downsampling-based initialization, can substantially improve the global structure of the final embedding because it survives through the optimization process. Importantly, a custom initialization does not interfere with t-SNE optimization and does not yield a worse solution compared to a random initialization used by default. Figure 3AB. A possible concern is that a custom initialization might bias the resulting embedding by injecting some artifact global structure. However, if anything can be seen as injecting artifactual structure, it is rather the random initialization. The global arrangement of clusters in a standard t-SNE embedding often strongly depends on the random seed. We also showed that using large perplexity values, 1% of the sample size, substantially larger than the commonly used ones, can be useful in the SNE context. Our experiments suggest that whereas PCA initialization helps preserving the macroscopic structure, large perplexity, either on its own or as part of a perplexity combination, helps preserving the mesoscopic structure. Figure 3D, F. It has recently been claimed that UMAP preserves the global geometry better than t-SNE 11. However, UMAP operates on the nearest neighbor graph exactly as t-SNE does, and is therefore not designed to preserve large distances any more than t-SNE. To give a specific example, Ref. 8 performed both t-SNE and UMAP, and observed that unlike t-SNE, UMAP places related cell types near one another. We demonstrated that this is largely because t-SNE parameters were not set appropriately. Simply using high learning rate places related cell types near one another as well as UMAP does, and additionally using exaggeration factor separates clusters into more compact groups similar to UMAP. t-SNE is often perceived as having only one free parameter to tune, perplexity. Under the hood, however, there are also various optimization parameters, such as the learning rate, the number of iterations, early exaggeration factor, etc., and we showed above that they can have a dramatic effect on the quality of the visualization. Here we have argued that exaggeration can be used as another useful parameter when embedding large datasets. In addition, while the low-dimensional similarity kernel in t-SNE has traditionally been fixed as the t-distribution width degree of freedom, we showed in a parallel work that modifying can uncover additional fine structure in the data 41. One may worry that this gives a researcher too many knobs to turn. However, here we gave clear guidelines on how to set these parameters for effective visualizations. As argued above and in Ref 15, setting the learning rate to ensures good convergence and automatically takes care of the optimization issues. Perplexity should be left at the default value 30 for very large datasets, but can be combined with, for smaller datasets. Exaggeration can be increased to 4 for very large datasets, but is not needed for smaller datasets. In comparison, UMMAP has two main adjustable parameters and many further optimization parameters, n_neighbors corresponding to perplexity and min_dist controlling how tight the clusters become. The latter parameter sets the shape of the low-dimensional similarity kernel 10 and is therefore analogous to mentioned above. Our experiments with UMMAP suggest that its repulsive forces roughly correspond to t-SNE with exaggeration. 4. Supplementary figures 2, 3. Whether this is desirable depends on the application. With t-SNE, one can choose to switch exaggeration off and, for example, use the embedding shown in Figure 7c instead of Figure 7b. Several variants of t-SNE have been recently proposed in the literature. One important example is parametric t-SNE, where a neural network is used to create a mapping from high-dimensional input space to 2 dimensions and is trained using standard deep learning techniques to yield an optimal t-SNE result 42. Parametric t-SNE has been recently applied to transcriptomic data under the names net-SNE 43 and SQUIS 44. The latter method combined parametric t-SNE with a variational autoencoder and was claimed to yield more interpretable visualizations than standard t-SNE due to better preserving the global structure. Indeed, the network architecture limits the form that the mapping. This implicit constraint on the complexity of the mapping prevents similar clusters from ending up in very different parts of the resulting embedding. Also, in this approach, the most appropriate perplexity does not need to grow with the sample size as long as the mini-batch size remains constant. By default, SQUIS uses mini-batch size of 512 and perplexity 10, which likely corresponds to the effective perplexity of 2% of the sample size, similar to our 1% suggestion here. Another important development is hierarchical t-SNE, or HSNE 45. The key idea is to use random walks on the nearest neighbors graph of the data to find a smaller set of landmarks, which are points that can serve as representatives of the surrounding points. In the next round, the nearest neighbors graph on the level of landmarks is constructed. This operation can reduce the size of the dataset by an order of magnitude and can be repeated until the dataset becomes small enough to be analyzed with t-SNE. Each level of the landmarks hierarchy can be explored separately. Ref. 18 successfully applied this method to mass cytometry datasets with up to 15 million cells. However, HSNE does not allow to embed all points in a way that would preserve the geometry on the level of landmarks. Our approach to preserving global geometry of the data is based on using PCA initialization and large perplexity values. It can fail if some aspects of the global geometry are not adequately captured in the first 2 PCs, or by the similarities computed using a large perplexity. This may happen when the dataset contains very isolated but rare cell types. Indeed, a small isolated cluster might not appear isolated in the first 2 PCs because it would not have enough cells to contribute much to the explained variance. At the same time, large perplexity will make the points in that cluster be attracted to some arbitrary, unrelated clusters. As a result, a small cluster can get sucked into the middle of the embedding even if it is initialized on the periphery. This is what happened in the Shakar et al. Data set, Figure 4b. Cone and rod photoreceptor, yellow, and a macron cell, cyan clusters, ended up in the middle of the embedding despite being very different from all the bipolar cell clusters. This can be seen in the MDS embedding of the cluster means, which is unaffected by the relative abundances of the clusters. Thus, when t-SNE is done together with clustering, we recommend to supplement a t-SNE visualization with a MDS visualization of cluster means, as in Figure 2a. Alternatively, one could use PAGA46, a recent method specifically designed to visualize the relationships between clusters in RNA-seq data. This example highlights that our approach is not a final solution to preserving the global structure of the data. A principled approach would incorporate some terms ensuring adequate global geometry directly into the loss function while making sure that the resulting algorithm is scalable to millions of points. We consider it an important direction for future work. In the meantime, we believe that our recommendations will strongly improve t-SNE visualizations used in the current single-cell transcriptomic studies and may be useful in other application domains as well. Methods. The t-SNE loss function. The t-SNE algorithm 9 is based on the SNE framework 47. SNE introduced a notion of directional similarity of point-to-point. 1. Defining for every given point a probability distribution over all points. All are set to 0. The variance of the Gaussian kernel is chosen such that the perplexity of this probability distribution, 2, has some pre-specified value. The larger the perplexity, the larger the variance of the kernel, with the largest possible perplexity value equal to, corresponding to, and the uniform probability distribution is the number of points in the dataset. Importantly, for any given perplexity value, all but nearest neighbors of point will have very close to 0. For mathematical and computational convenience, symmetric SNE define unidirectional similarities. 3. Such that, i.e., this is a valid probability distribution on the set of all pairs. The main idea of SNE in its modifications is to arrange points in a low-dimensional space such that the similarities between low-dimensional points match as close as possible in terms of the Kullback-Leibler divergence. The loss function is thus 4. The main idea of t-SNE was to use a t-distribution with 1 degree of freedom, also known as Cauchy distribution, as the low-dimensional similarity kernel. 5. Where are low-dimensional coordinates, and as a matter of definition, we consider any method that uses the t-distribution as the output kernel and Kullback-Leibler divergence as the loss function to be t-SNE similarities can in principle be computed using non-Euclidean distances instead of, or can use non-perplexity-based calibrations. To justify our intuitive explanation in terms of attractive and repulsive forces, we can rewrite the loss function as follows. 6. And dropping the constant. 7. To minimize. The first sum should be as large possible, which means large, i.e., small, meaning an attractive force between points. And, whenever, at the same time, the second term should be as small as possible, meaning small, and a repulsive force between any 2 points. And, independent of the value of, the t-SNE optimization. The original publication 9 suggested optimizing, using adaptive gradient descent with momentum. The authors initialized, randomly, using a Gaussian distribution with standard deviation 0.0001. It is important that initial values have small magnitude, otherwise optimization often fails to converge to a good solution. To escape bad local minima and allow similar points to be quickly pulled together, the original publication 9 suggested an early exaggeration trick. During initial iterations, they multiplied all attractive forces by alpha greater than 1. Later work 32 used alpha equals 12 for the first 250 iterations, which became the default since then. The exact t-SNE computes similarities and pairwise attractive and repulsive forces on each gradient descent step. This becomes unfeasible for. Follow-up paper 32 suggested 2 approximations in order to speed up the computations. First, it noticed that for any perplexity value, all but nearest neighbors of any given point will have nearly 0 values. It suggested to only find nearest neighbors of each point in set. For the remaining points, This relied on finding the exact nearest neighbors, but in later work, various authors, 10, 16, 39, 48, started using approximate nearest neighbor algorithms, which is much faster and does not seem to make t-SNE results any worse. Using nearest neighbors accelerates computation of the attractive forces. To accelerate the repulsive force computations, Ref. 32 used the Barnes-Hutt approximation, originally developed for n-body simulations in physics. This reduces computational complexity from 2. Works reasonably fast for up to, but becomes too slow for much larger sample sizes. Inspired by the fast multipole method, another technique originally developed for n-body simulations, Ref. 16 suggested to interpolate repulsive forces on an equispaced grid and to use fast Fourier transform to accelerate the interpolation, t-SNE. This lowers computational complexity too, and works very fast for into millions. An important limitation is that it is only implemented for 1D and 2D, but not for 3D embeddings, as the interpolation would work much slower in 3D. Synthetic dataset. All points were generated from a standard Gaussian distribution in 50 dimensions. All points from each class were shifted by 20 in mutually orthogonal directions. All points from each type in one class, per type, were additionally shifted by 4 in mutually orthogonal directions. In another class, per type, by 10. In the third class, per type, also by 10. The resulting total sample size was dot. Data preprocessing. All datasets were downloaded as tables of UMI or red counts. Let be a matrix of gene counts with and being the number of cells and the number of genes respectively. We assume that 0 columns, if any, have been removed. We used a standard preprocessing pipeline consisting of the following steps: I. Sequencing depth normalization. 2. Feature selection. 3. Log transformation. 4. PCA. Specifically, we normalized the read counts to counts per million CPM and UMI counts to counts per median sequencing depth, selected 1,000 to 3,000 most variable genes using dropout-based feature selection similar to the one suggested in Ref 49, applied transform, and finally did PCA retaining the 50 leading PCs. We experimented with modifying and omitting any of these steps. Our experiments showed that log transformation, or a similar nonlinear transformation, and feature selection are the two most important steps for adequate results. PCA mainly improves computational efficiency as it reduces the dimensionality and size of the dataset before running t-SNE. Sequencing depth normalization. We divided the read counts of each cell by the cell's sequencing depth and multiplied by 1 million to obtain CPM. We normalized the UMI counts by the cell's sequencing depth and multiplied by the median sequencing depth across all cells in the dataset 50. This is more appropriate for UMI counts, because multiplying by 1 million can strongly distort the data after the subsequent transform 51. Feature selection. Most studies use the mean variance relationship to perform feature selection. They select genes that have large variance given their mean. We adopt the approach of Ref. 49 that exploited the mean dropout relationship. The idea is to select genes that have high dropout, i.e. zero count frequency, given their mean expression level. Any gene that has high dropout rate and high mean expression could potentially be a marker of some particular subpopulation. We found it more intuitive to use the mean across non-zero counts instead of the overall mean, because it is computed independently of the fraction of zero counts. For each gene, we computed the fraction of near zero counts, 8, and the mean log non-zero expression, 9. For all UMI-based datasets we used, and for all SMART-seq 2/smarter datasets we used. Some known marker genes were not getting selected with. We discarded all genes that have non-zero expression in less than. There was a strong negative relationship between and across all the remaining genes, supplementary figure 4. In order to select a pre-specified number of genes, usually, or we used a heuristic approach of finding a value, such that 10 was true for exactly. This can be done with a binary search. In supplementary figure 4, this corresponds to moving the red line horizontally until there are exactly genes to the upper right of it. These genes were then selected. We used for all datasets apart from Makosco et al., where provided a better fit for the distribution. We performed feature selection using the raw counts before sequencing depth normalization. Then we used normalized values for the selected genes. We used for the t-SNE et al. 2018 and for the Makosco et al. datasets, and for the remaining datasets. This feature selection method was not used for the 10x genomics and the Cow et al. datasets, see below. Nonlinear transformation. We transformed all values in the count matrix after feature selection with a transformation. This transformation is standard in the transcriptomics literature. It is convenient because all zeros remain zeros, and at the same time the expression counts of all genes become roughly comparable. Without this transformation, the Euclidean distances between cells are dominated by a handful of genes with very high counts. There are other transformations that can perform similarly well. In the cytometric literature, the inverse hyperbolic sign is often used, sometimes as with or a similar value 17. Note that is the variance stabilizing transformation for the negative binomial distribution with parameter, which is often taken to model UMI, counts well. Standardization. Many studies standardize the matrix after the log transformation, i.e., center and scale all columns to have zero mean and unit variance. We prefer not to do this by default. In general, standardization is recommended when different features are on different scale, but the log transform counts of different genes are arguably already on the same scale. From a more theoretical point of view, if one could assume that the expression counts of each gene for cells of the same type are distributed log normally, then Euclidean distance after log transformation would exactly correspond to the log likelihood. For the UMI-based data, the common assumption is that the expression counts are distributed according to the negative binomial distribution. For large counts, the negative binomial distribution behaves qualitatively similarly to the log normal. For example, its variance function is, whereas the log normal has variance function proportional to. So the Euclidean distance after the log transformation can be thought of as approximating the negative binomial log likelihood 26. Standardizing all the genes after log transformation would destroy this relationship. At the same time, in some datasets, we observed a stronger separation between some of the clusters after performing the standardization step. Here we applied standardization for those datasets in which it was used by the original authors, Makosco et al., Shakar et al., 10x genomics, Cow et al. Scampi preprocessing. For the 10x genomics and the Cow et al. datasets, we used the preprocessing pipeline recipe Discujung 17 from Scampi 23 to ease the comparison with clustering and dimensionality reduction performed by Ref 23 and Ref 8. This pipeline follows Ref and is similar to ours. It performs sequencing depth normalization to median sequencing depth, selects most variable genes based on the mean variance relationship, applies the transform and standardizes each feature. We used genes for the 10x genomics dataset and for the Cow et al. dataset following the original publication. Principal component analysis. We used PCA to reduce the size of the data matrix from 2 prior to running t-SNE. In our experiments, this did not have much influence on the t-SNE results, but is computationally convenient. Some studies, for example Ref 20, estimate the number of significant principal components via shuffling. In our experiments, for the datasets with tens of thousands of cells, the number of significant PCs was usually close to 50. For example, for the t-SNE et al. 3 dataset it was 40, according to the shuffling test. Given that PCA does not have much influence on the t-SNE results, we prefer to use a fixed value of 50. Default parameters for t-SNE optimization. Unless explicitly stated, we use the default parameters of fit-SNE. Following Ref 32, The defaults are 1000 iterations with learning rate. Momentum 0.5 for the first 250 iterations and 0.8 afterwards, early exaggeration. For the first 250 iterations, initialization drawn from a Gaussian distribution with standard deviation 0.0001. Further input parameters for the nearest neighbor search using the Annoy library, number of trees, 50, number of query nodes, for perplexity. And for the grid interpolation, number of approximating polynomials, 3, maximum grid spacing, 1, minimum grid size, 50, were always left at the default values. For reproducibility, we always used random seed 42. Initialization. For PCA initialization, we divide the first two principal components by the standard deviation of PC1 and multiply them by 0.0001, which is the default standard deviation of the random initialization. This scaling is important. Values used for initialization should be close to 0, otherwise the algorithm might have problems with convergence. We learned about the importance of scaling from James Melville's notes at https://melville.github.io/smallvis/init.html. The same scaling was used for the downsampling-based initialization and also for the custom initialization when creating aligned visualizations. The sign of the principal components is arbitrary. To increase reproducibility of the figures, we always fixed the sign of the first two PCs such that for each PCA eigenvector, the sum of its values were positive. Post-processing. For the t-SNE embeddings of the 10x genomics dataset, we rotated the result 90 degrees clockwise and flipped horizontally to make it visually more pleasing. Note that t-SNE result can be arbitrarily rotated and flipped, as this does not change the distances between points. Caveat: it should not be stretched horizontally or vertically. Multi-scale similarities. We follow Ref 13 in the definition of multi-scale similarities. For example, to combine perplexities 30 and 300, the values are computed with perplexity 30 and with perplexity 300 for each cell, and then averaged. This is approximately, but not exactly, equivalent to using a different similarity kernel instead of the Gaussian kernel. Where is Euclidean distance a multi-scale kernel? 11, with the variances, and selected such that the perplexity of the first Gaussian component is 30 and the perplexity of the second Gaussian component is 300. Learning rate. Following Ref 15, we used learning rate as it is defined in fit-SNE. Fit-SNE, as well as OpenSNE, followed the convention of the original 9 and the Barnes-Hut 32 t-SNE implementations and omitted factor 4 in the gradient equation. Some other existing t-SNE implementations, such as the one in scikit-learn, do include the factor 4. There one would need to use to achieve the same result. Exaggeration. Early exaggeration means multiplying the attractive term in the loss function, EQ7, by a constant. During the initial iterations, the default is for 250 iterations. Ref 34 suggested to use late exaggeration, using sum, for some number of last iterations. Thus, their approach used 3 stages: early exaggeration, followed by no exaggeration, followed by late exaggeration. We prefer to use 2 stages only. We keep constant after the early exaggeration is turned off. This is why we simply call it exaggeration and not late exaggeration. While early exaggeration is essentially an optimization trick 9, we consider subsequent exaggeration to be a meaningful change of the loss function. Positioning out of sample cells. To position new cells on an existing t-SNE embedding, we use the same. Most variable genes that were used to create the target embedding, usually only a subset of genes was present in the count table of the new dataset. We used transformed counts of these genes to compute the correlation distances. To position a cell, we used coordinate-wise median among its nearest neighbors. We used correlation distances as possibly more robust for batch effects than Euclidean distances. When out-of-sample cells are sequenced with a different protocol, the batch effect can be very strong. This consideration does not apply to positioning cells for a downsampling-based initialization without any possible batch effect. For computational simplicity, here we used Euclidean distance in the space of the 50 PCs. Bootstrapping over genes. We used bootstrapping to estimate the uncertainty of the mapping of new cells to the existing t-SNE atlas. Given a set of genes that are used for the mapping, we selected a bootstrap sample of genes out of with repetition and performed the positioning procedure using this sample of genes. This constitutes one bootstrap iteration. We did 100 iterations and for each cell obtained 100 positions on the t-SNE atlas. The larger the spread of these positions, the larger the mapping uncertainty. For Figure 5c, we computed the distances from the original mapping position to the 100 bootstrap positions and discarded 5 bootstrap positions with the largest distance. Then we drew a convex hull of the remaining 95 bootstrap positions, using scipy.spatial.convex hull. Reporting summary. Further information on research design is available in the Nature Research Reporting Summary linked to this article. Data availability. All data were downloaded in the form of count tables following links in the original publications. Code availability. We prepared a self-contained Jupyter notebook in Python that demonstrates all the techniques presented in this manuscript. It is available at https://github.com/berenslab/RNA-seq. For simplicity, it uses the Tosic et al. .3 dataset for all demonstrations. To show how to map new cells to the reference t-SNE atlas, we split the data into a training set and a test set. To show how to align two t-SNE visualizations, we split the dataset into two parts. To show how to process a much larger dataset, we replicate each cell 10 times and add noise. The code that does the analysis and produces all the figures used in this manuscript is available in form of Python notebooks at https://github.com/berenslab/RNA-seq. We used a C++ implementation of fit-SNE 16, version 1.1, available at https://github.com/klugerlab/fit-SNE, together with interfaces for R, MATLAB, and Python. While working on this paper, we contributed to this package several additional features that were crucial for our pipeline. Fit-SNE has been re-implemented as a pure Python package, OpenSN52, available at https://github.com/pavlinpolicar/OpenSN. It supports all the features used in this manuscript and conveniently allows us to position out-of-sample cells with or without optimization. References. Sandberg, R. Entering the Era of Single-Cell Transcriptomics in Biology and Medicine. Nat Methods 11, 22-2014. Poulin, J. F. Tosic B. Gerling-Leffler, J. Trimarchi J. M. and Altramani R. Disentangling Neural Cell Diversity Using Single-Cell Transcriptomics. Nat Neuroscience 19, 1131, 2016. Tosic, B. et al. Shared and Distinct Transcriptomic Cell Types Across Neocortical Areas. Nature 563, 72, 2018. The Tabula Muris Consortium. Single-cell transcriptomics of 20 mouse organs creates a tabula muris. Nature 562, 367-372, 2018. Seisel, A. et al. Molecular Architecture of the Mouse Nervous System. Cell 174, 999-1014, 2018. Hahn, X. et al. Mapping the Mouse Cell Atlas by Microwell-6 Cell 172, 1091-1107, 2018. Saunders, A. et al. Molecular Diversity and Specializations Among the Cells of the Adult Mouse Brain, Cell 174, 1015-1030, 2018. Cow, J. et al. The Single-Cell Transcriptional Landscape of Mammalian Organogenesis, Nature 566, 496, 2019. Van der Motten, L. and Hinton, G. Visualizing Data Using t-SNE JMOC, Learning Res 9, 2579-2605, 2008. Math. McInnes, L., Healy J. and Melville J. Map. Uniform Manifold Approximation and Projection for Dimension Reduction, https://archive.org/ab/1802.03426, 2018. Becht, E. et al. Dimensionality Reduction for Visualizing Single-Cell Data Using UMAP. Nat Biotechnol 3738, 2019. Wattenberg, M., Viegas, F. and Johnson, I. How to Use t-SNE Effectively. Distill, http://distill.pub/2016/misread 2016. Lee, J.A., Pelifo Ordóñez, D.H., and Verleeson, M. Multiscale Similarities in Stochastic Neighbor Embedding, Reducing Dimensionality while Preserving Both Local and Global Structure. Neurocomputing 169, 246-261, 2015. Bott, C.D., Mulders D., Verleeson M., and Lee J.A., Perplexity-Free t-SNE, and Twice Student t-SNE. In European Symposium on Artificial Neural Networks, Computational Intelligence and Machine Learning, 123-128, 2018. Belkina, A.C. et al. Automated Optimized Parameters for T-Distributed Stochastic Neighbor Embedding Improve Visualization and Allow Analysis of Large Datasets. NatComs, https://doi.org/10.1038/S414670019 to 13055 year, 2019. Linderman, G.C., Rack M., Hoskins, J.G., Steinerberger, S. and Kluger, Y. Fast Interpolation-Based t-SNE for Improved Visualization of Single-Cell RNA-seq Data. Nat Method 16, 243, 2019. Amir, E.D. et al. Visens enables visualization of high-dimensional single-cell data and reveals phenotypic heterogeneity of leukemia. Nat Biotechnol 31, 545, 2013. Unen, V. et al. Visual analysis of mass cytometry data by hierarchical stochastic neighbor embedding reveals rare cell types. Nat Coman 8, 1740, 2017. Lee, W. Cerise, J. E., Yang, Y. and Han, H. Application of t-SNE to Human Genetic Data. J. Bioinform, Compute Bio 15, 1,750,017, 2017. Diaz Popkovich, A. Anderson Trochme, L. Gravel, S. Revealing Multi-Scale Population Structure in Large Cohorts, https://www.bioarxiv.org/content/10.1101/423632v2, 2018. Schmidt, B. Stable Random Projection, Lightweight General-Purpose Dimensionality Reduction for Digitized Libraries, http://culturalanalytics.org/2018/09/Stable Random Projection Lightweight General-Purpose Dimensionality Reduction for Digitized Libraries/2018. Lee, J. A. and Verleeson M. Quality Assessment of Dimensionality Reduction, Rank-Based Criteria. Neurocomputing 72, 1431-1443, 2009. Wolf, F.A., Angerer, P. and Thace, F.J. Scampi, Large-Scale Single-Cell Gene Expression Data Analysis, Genome Bio 19, 15, 2018. McCosco, E.Z. et al. Highly Parallel Genome-Wide Expression Profiling of Individual Cells Using Nanoliter Droplets, Cell 161, 1202-1214, 2015. Shakar, K. et al. Comprehensive Classification of Retinal Bipolar Neurons by Single-Cell Transcriptomics, Cell 166, 1308-1323, 2016. Harris, K. D. et al. Classes and continua of hippocampal CA1 inhibitory neurons revealed by single-cell transcriptomics. Plos Bio 16, E2006387, 2018. Cadwell, C.R. et al. Electrophysiological, transcriptomic, and morphologic profiling of single neurons using PatchSec Nat Biotechnol, 34, 199, 2016. Kiselyov, V.Y., U.A. and Hemberg, M. SCMAP. Projection of single-cell RNA-seq data across datasets. Nat Methods 15, 359, 2018. Berman, G.J., Choi D.M., Bialek, W. and Shavitz, J.W. Mapping the Stereotyped Behavior of Freely Moving Fruit Flies, J. Roy Sok, Interface 11, 20,140,672, 2014. Polakar, P.G., Strasser, M. and Zupin, B. Embedding to reference t-SNE space addresses batch effects in single-cell classification, https://www.bioxiv.org/content/10.1101/671404v1, 2019. Tosich, B. et al. Adult Mouse Cortical Cell Taxonomy Revealed by Single-Cell Transcriptomics, Nat Neurosci 19, 335, 2016. Van der Matten, L. Accelerating t-SNE Using Tree-Based Algorithms, J. Mach, Learning Rees 15, 3221 to 3245, 2014. Math SciNet. Math. Linderman, G.C. and Steinerberger, S. Clustering with t-SNE Provably. Siam J. Math, Data Sci 1, 313 to 332, 2019. Math SciNet. Linderman, G.C., Rack M., Hoskins, J.G., Steinerberger, S. and Kluger, Y. Efficient Algorithms for T-Distributed Stochastic Neighborhood Embedding, https://archive.org/ab/1712.09005, 2017. England, C. et al. Pack 6, TBR2 and TBR1 are expressed sequentially by radial glia, intermediate progenitor cells, and postmitotic neurons in developing neocortex. J. Neurosci 25, 247 to 251, 2005. Yuzwa, S.A. et al. Developmental Emergence of Adult Neural Stem Cells as Revealed by Single-Cell Transcriptional Profiling. Cell Rep 21, 3970 to 3986, 20.